Okay, I just want to start by giving my thanks to the Joint Genome Institute, and especially to the Community Sequencing Program, which is a really fantastic thing. I know I've heard from several of the speakers in the past day and a half that sequencing is now free. But apparently nobody's told our accounting department about that. They keep sending me bills, and so I'm really appreciative of the fact that the Community Sequencing Program is here so that a small lab like mine can do things that we never really imagined that we could do otherwise. Okay, so with that, let me get started. I told you I'd talk about how to eat a wooden ship, and so I think it's probably safe to say that there are not a whole lot of organisms on the planet that would consider this to be a food item. But I want to talk to you about a group of organisms that do exactly that. They're called shipworms, and they're kind of a worm-like marine mollusk that makes its living by burrowing into wood in marine environments, and then they actually eat the wood as their primary source of nutrition. So shipworms are actually voracious consumers of wood, and they cause billions of dollars of damage to marine structures, including ships and piers and various types of fishing gear and aquaculture enclosures. We call them shipworms, but actually the name is a misnomer. They're not actually worms at all. Instead, they are really clams. And they're quite closely related to clams like this one that we're really familiar with. That's the steamer clam, Maya arenaria. That's a pretty popular food item in New England, where I come from. And it's the closest relative, one of the closest relatives of the shipworms. So you can see, here's a shipworm. This is Lyrotis pedicellata. So you can see it doesn't look very much like a clam, but it is. And you can tell that because it has a pair of shells located here. But unlike other bivalves that use their shells as a form of protection, in the shipworm, the shell is a drill bit. And the surface of the shell is covered with tiny little teeth. And it uses those teeth to grind its way into the wood. So you can see here it's burrowing into the wood. And then it ingests the wood particles as it burrows, and that becomes its major source of nutrition. So before I go on, I just want to point out a couple of pieces of shipworm anatomy here. Here is where the mouth of the shipworm is. So when the shipworm burrows, it eats the wood particles, and then they go into this big sac here. You can see it's white because it's full of wood particles. The other thing I want to point out is that the rest of the animal here is filled up with a large gill. So the gill is one of the largest organs in the shipworm. It takes up about 30% of the animal's mass. So I want you to remember that because it turns out that the gills figure importantly in the shipworm's biology. So just keep that in mind as we go along. Okay, so shipworms are common. They're quite diverse, and they're widespread throughout the world's oceans. There are about 65 species, and they can be found burrowing in all kinds of wood. And not only just wood, but they also burrow in a variety of different types of woody or fibrous plant materials. For example, some species are adapted for growing for burrowing in the rhizomes of seagrass. Some can be found burrowing in hemp rope or cork. There are even some that will burrow into the shells of coconuts. So you can guess from that that shipworms are capable of degrading a really broad variety of plant materials. And they're not terribly picky about what they eat. So as you probably guessed, there's not many animals in the world that can eat wood. Basically on land it's termites, and in the oceans, shipworms are the primary wood consumers. The reason is that wood is pretty hard to digest. And that's because it's made from lignocellulose, which is really a pretty remarkable material. It's a composite of cellulose fibers embedded in a matrix of hemicellulose and lignin. And this is a material that has evolved over hundreds of millions of years of natural selection to be incredibly strong and light. Also, it's insoluble and very resistant to enzymatic decay. However, of course, there's a lot of interest now in figuring out how to break down cellulose with enzymes, and that's because of the growing interest in cellulosic waste material as a possible renewable energy source. So the idea here is to take cellulosic materials that you could find in things like agricultural or domestic paper waste or even forest product waste, break it down into its component sugars, and from there make alcohol that you can use for transportation fuel. So this part of the process is easy. That's fermentation, and of course that's one of the oldest scientific technologies known to man and probably the best loved. But this part of the process is the hard part. It turns out the bottleneck is that we don't know of enzymes that can efficiently and inexpensively degrade cellulose to its component sugars. Now, just a word about the enzymes. The enzymes that break down lignocellulose belong to a group that's called Kzymes, or carbohydrate active enzymes. So if I use that term Kzymes, you'll know what I mean. These are modular enzymes. They can often have multiple catalytic domains and multiple cellulose binding modules. So the binding modules serve to attach the catalytic domains, or catalytic modules, to their insoluble substrate. There are 5 different groups of Kzymes, but I'm only going to talk about one of them. Those are the glycoside hydrolases. And if I forget, I'll probably call these GH modules, so you'll know what I'm talking about when I say that. They're glycoside hydrolases. And this is the group that's responsible for most of the catalytic modules that break down lignocellulose. Okay, so I've said that lignocellulose is a complex material, and as a result of that, it takes a fairly large variety of enzymes to reduce it to its component sugars. So the simplest component is glucose, and even this, which is just a simple linear polymer of glucose, takes 3 different classes of enzymes to break it down. Hemicellulose is still more complex. It's a branched chain molecule with lots of different 5 and 6 carbon molecules substituting on it. And this takes a correspondingly larger number of enzymes to completely deconstruct it. So in all, over 20 different types of catalytic activities have been identified to be important in the breakdown of lignocellulose. So the take-home message here is that in nature, cellulose is not degraded by a single enzyme. It's degraded by a whole cocktail, a complex cocktail of different enzymes with different specificities, all working together to break down this complex material. And as a consequence of that, we rarely find any single microorganism that has all of these enzymes. So in nature, when you see lignocellulose degradation occurring, it's happening it's accomplished by a complex consortium of microbes. So for example, if you look in the gut of a termite, you'll find that there are over 1,000 different microbial types. Here, if you kind of squeeze out the goo inside of the hindgut of the termite. And they're both protozoa and bacteria, so it's quite a complex community. The same thing is true of ruminants. So here's my highly artistic drawing of a ruminant. So ruminants break down cellulose. Actually, pretty much all animals that have been described to date that can use cellulose as a food do it in pretty much the same way. So here the cow ingests plant matter that's rich in cellulose, goes into its stomach, which is basically a sort of complicated anaerobic fermenter with lots of different chambers and compartments. In there are over 1,000 different types of bacteria that attach themselves to the substrate and help to break it down. The shipworm digestive tract is completely different. It's much simpler than the cow rumen. And in fact, we can model it very simply this way. There are just 2 components, this large sac-like cecum and then a relatively short and straight intestine. So wood particles go into the cecum, pass through the intestine, and then out as frass. Frass is our word for shipworm poop. Okay, so we've been able to show that lignocellulose degradation occurs in both of these chambers, and you can show that with carbohydrate analysis. So here we're just looking at one of the carbohydrates. This is glucan, which is the major component of cellulose. And you can see what you can do is compare the wood which starts out with a glucan concentration of about 40%. But once that wood enters the cecum, it drops to about 30%. And then by the time the material exits as frass, it's down to about 20% glucan. So we can see that there's pretty substantial, oh, how about that? I forgot the arrows. There's pretty substantial degradation of cellulose during the passage through the shipworm gut. So now we can add to the model that we know that there's lignocellulose active enzymes in both the cecum and the intestine. So the next question is then, where do these enzymes come from? If this were a termite or a ruminant, these would come predominantly from thousands of different bacterial types that are present in those compartments. But the shipworm is a completely different story. It turns out that the shipworms have almost no bacteria in their cecum. So you can see this in some work that was done by one of the students in our project, Megan Betcher. She's from Oregon Health Sciences University and is part of a project that we have as a DOE-funded project looking at the symbionts of mollusks in the Philippines. It's called the PMS-ICBG project. So what she's done is she's taken a section of the shipworm tissue right about here where it intersects both the gill, which is up here in this region, and the cecum, which is down here. And I don't know if you can see that, but there's a lot of chips of wood in there. And what she's done is she's hybridized this with a probe, a DNA probe, that will hybridize to pretty much all bacteria. And that probe is labeled with a red fluorescent molecule. So what you expect here is that wherever there are bacteria, you should see red fluorescence. Now, what she found was that when you look in the cecum, and this is the probe, and there's the mismatch negative control, you see very few bacteria. You might see one or two. I think there's one down there. You might not be able to see it. But the bacteria are extremely rare in there. However, you know, the 800-pound gorilla in the room here is up here. This is the gill, and you notice that it lights up like a Christmas tree. So if we look at that more closely, you see that these cells, you see these amorphous cells inside the gill, and those turn out to be chock full of bacteria. So this is an electron micrograph, and you can see the cytoplasm of the cell here, and there's sort of a granular material there. And if you look at that more closely, you see it's completely made up of these long gram-negative rods. So there's lots of bacteria in the gill. So what type of bacteria are they? A number of years ago, one of my early mentors, John Waterbury from Woods Hole Oceanographic Institution, isolated this bacterium. He's called Pteridinobacter turneri, and found that he could find it in the gills of shipworms from all around the world, many different shipworm species. It turns out that Pteridinobacter is a gram-negative gamma proteobacterium, and it's a really good cellulose degrader. Here you can see what it does to filter paper. Filter paper is 99% cellulose. So this is filter paper in sterile seawater. It's inoculated on day 0. By day 14, there's no trace of the filter paper left. Okay, so for a long time, we thought this guy, this is Pteridinobacter up there, was the sole symbiont of shipworms. But once we started looking at shipworms using molecular methods, we discovered that there are multiple shipworm symbionts, and this tree shows some of the different ribotypes that we've found. So it's a consortium, much like the termite and ruminant guts, but it's different in some really important respects. First of all, the symbionts are intracellular, so they're actually inside the host cells. They're not in the gut, which is essentially the outside world. Also, it's a much, much simpler consortium than you find in termites or ruminants. There are only about 10 symbiont types, as opposed to over 1,000 for either termite or ruminant. The symbionts are all very closely related to one another, whereas in the termites and ruminants, they're drawn from many different bacterial phyla. These are 95% related at the 16S level or more, or closer. And they're unrelated to anything that's been identified in other cellulose-degrading systems. Now, the picture we had for a long time was that this was an easy bacterium to cultivate, but the rest of these we never had any luck in cultivating. But just in this past year, we had a student visit from the Philippines, from the University of Philippines, Marvin Altamia, and this is also funded by the PMS-ICBG. And Marvin turned out to have a really great green thumb for growing these bacteria, and he was able to isolate 5 different strains that correspond with some of the more abundant ribotypes that we found in the gills. So this is a pretty good breakthrough for this model system, because it's rare to be able to have symbionts that grow in pure culture. In fact, I don't think anybody has as many as we do. So now we can add another couple of pieces to the puzzle. The cecum is bacteria-free. However, there's another extremely large reservoir of bacteria in the shipworms, but it's not in the gut at all. It's in a separate organ called the gill, and you saw how big that gill is. And we also know that those bacteria are capable of producing cellulolytic enzymes. So the big question is whether these bacteria can produce enzymes and somehow transport them to the gut to aid in cellulose degradation. So if this were in fact the case, it would be a pretty fascinating model system. So there's a lot of advantages to this system. It's very low complexity. There are very few symbiont types. That makes it highly amenable to genomics and proteomics. Both the host and symbionts are phylogenetically distinct from anything that's been studied before. The anatomy tells us that the system functions very differently from other lignocellulose degraders. And finally, both the animals and now several of the major symbiont types can be grown in the lab in culture. Okay, so now we have a pretty interesting model system that we can begin to explore and to test using genomic and proteomic methods. So we started with the obvious. We took the gills out of a shipworm, Bankia setacea, and sequenced the gill symbiont metagenome and also the genomes of so far 3 of these symbionts, Pteridinobacter plus 2 of those new isolates that belong to the group that is quite difficult to cultivate. So by doing this, we can now compare the metagenome, which contains the total portfolio of genes from the symbiont community, and then we can compare that to the genomes of the individual isolates. And we can start to ask interesting questions like what genes are present in the metagenome and in the isolates, and, oops, jumped ahead a little there, and we can ask whether the symbiont isolates differ from one another, whether they are contributing something different to the symbiosis or whether they're all doing the same thing. We can also compare the shipworm gill metagenome to some of the other metagenomes that have been sequenced recently, like the termite hindgut and the cow rumen. And finally, we can use these genomes as a database for genome-enabled proteomics. So we can actually begin to ask which of these proteins are actually expressed in the system and might actually participate in the symbiosis. So to start with, we'll look at some of the isolates, and you can see immediately, just from the genome statistics, that BSO2 and BSO8, which are these 2 new isolates, are quite a bit smaller than Pteridinobacter. So BSO2 has 3.7 million base pairs in its genome, BSO8 4.8, and T. terneri 5.2. So there's about a 30% difference in size of the genomes from BSO2 to Pteridinobacter. And that's reflected in the number of protein-coding genes that you observe in those genomes. You can also see that these genomes are really rich in Kzymes, in the carbohydrate active enzymes. So this is just looking at those glucoside hydrolase genes. They range from 86 to 103. And that's a pretty substantial proportion of the protein-coding genes in the genome, ranging from about 2.3 to 2.6% of protein-coding genes. So that's a pretty rich source of cellulose-degrading genes. And finally, we've only had these genomes for a short amount of time, but we've been able to spot some obvious metabolic differences between these organisms. So for example, BSO2 and BSO8, we predicted both were able to grow anaerobically with nitrate, so they're nitrate respirers, whereas Pteridinobacter lacks those genes. Similarly, BSO8 and T. terneri have genes for nitrogen fixation, but BSO2 does not. And we were actually able to confirm these predictions with culture experiments. So the genomes of these isolates are quite different from one another. But the next question is, how do they stack up as cellulose degraders? So to try to get a preliminary idea of that question, we compared them to what I call the enzyme champs. These are the organisms that have been identified that encode the most enzymes for the breakdown of carbohydrates. And the all-time grandmaster is Bacteroides theta iota micron. This encodes a whopping 260 GH or glucosyl hydrolase modules. The other one we're comparing to is Saccharophagous degradans. That's known as one of the most capable degraders of complex polysaccharides, and it encodes about 131. So you can see that the shipworm bacteria encode a lot, but it's not even close to some of the enzyme champs. But you get a little bit different picture when you just look at the glucoside hydrolase genes that function against cellulose and hemicellulose. They're shown in green here. And now what you see is anywhere from 50 to 60% of the K-zome, as the total number of GH molecule proteins, are targeted against cellulose or hemicellulose, whereas in S. degradans it's only about 29% and a measly 5% in B theta. So actually it turns out that these guys have more GH modules acting against cellulose. That means they're more specialized for that particular substrate. So now we can ask the question whether these bacteria are outliers or whether they're typical of, they have the typical characteristics of the metagenome. And in fact, it turns out that they are typical of what we see in the metagenome. We sequenced about 26 million base pairs in the metagenome, and that allowed us to call about 40,000 proteins. About half of them have functional predictions, and from those, about 1,000 are K-zymes. So again, that's very rich in K-zymes. Out of those 1,000, 600 are active against cellulose and hemicellulose. So well above 1% of all of the protein-coding genes in this metagenome are active against cellulose and hemicellulose. That's very rich. And we can then do the same sort of exercise. We can compare them to the metagenome K-zyme champs. So here, the all-time K-zyme champ is the cow rumen. It encodes over 12,000 GH modules. The termite comes in a distant second with 700, and the shipworm's a close third with about 500. But again, if you look at it in terms of the fraction that are active against cellulose and hemicellulose, the shipworm's the winner again. About half of its GH modules are active against cellulose and hemicellulose, whereas only about a quarter of the termite and the rumen are active against that. Now, this guy obviously wins for total numbers, so this is sort of the mother lode. But by the same analogy, the shipworm is the rich vein. It's a very concentrated source of enzymes of interest. So now we know that the metagenome is at least potentially very capable of degrading cellulose. So the next question is, which, if any, of these proteins are actually expressed in the symbiosis? And to do that, we did that by a proteomic method called 2D-LC-MSMS. And so basically what you do here is we extract out the gill and the cecum from the shipworm. And we extract protein from them. The gill tissue we know is full of bacteria. The cecum is symbiont-free. Then we separate the proteins by liquid chromatography. That's the LC part. We digest them with a protease, and then measure the size of the peptides that you get using mass spec. You can then take the metagenome data and do an in silico digest. That is, predict what peptides masses would be produced if you digested them with the same protease. Then by comparing the predicted peptide masses to the measured peptide masses, you can identify proteins, proteins that are in these samples. So this method is really cool because it only sees the proteins that are encoded in the symbiont metagenome. So we don't see the host proteins, we don't see proteins that might come from, say, other microorganisms. And so with this method, we can ask that burning question that we've wanted to ask all along, which is, can we find symbiont-encoded proteins in the symbiont-free cecum? And if we can, that means that those proteins are somehow getting from the gill to the gut. If we can see them, we can ask which proteins are they and what might they be doing in the symbiosis. So when we actually did this experiment, we got some really interesting results. We measured 29 proteins, or we detected 29 proteins that we could only find in the cecum, a similar number, 44 proteins that we could only find in the gill, and an overlap of 9 proteins that were in both compartments. So that tells us that we can indeed detect symbiont-encoded proteins in the cecum. So those proteins are somehow getting from the gill to the cecum, but the question is how. And we could also say that we see very little overlap between the proteins in the cecum and the proteins in the gill. So these are very different communities of protein. Now the interesting thing happens when you actually look at what these proteins are. So in the cecum, it turns out that 27 of those 29 proteins are carbohydrate-active proteins. Whereas in the gill, we don't see, in the gill-only fraction, we don't see any Kzymes. There are 3 Kzymes that are found in both compartments. So if you look at the gill-only fraction, you see that these are exactly the proteins you would expect to find in a tissue full of these bacteria. They're all these structural proteins and proteins of core metabolism that you'd expect to be abundant in bacterial cells. But if you look in the cecum, it's very different. We don't see any of those housekeeping genes. Instead, we only see carbohydrate-active enzymes, and only those carbohydrate-active enzymes that are active against cellulose and hemicellulose. And you can see there's a broad selection of types of enzymes, basically all the types of enzymes you'd expect to want to have to break down cellulose and hemicellulose. So you've got endoglucanases, cellobiohydrolases, endoxylinase, xylene esterases, and a variety of cellulose binding modules that specifically target cellulose and hemicellulose. So this is a pretty remarkable result, because what it tells us is that lignocellulose active enzymes are selectively transported to the gut from the gill to the exclusion of all those other proteins that we know are abundant in those bacteria. This is pretty amazing. We don't really know of any other organism that does anything like this, cellulolytic or not. So now we can answer this burning question that we've always wanted to answer with a resounding yes. Enzymes produced by these intracellular bacteria in the gill are somehow transported by a mechanism that we don't understand into the gut, where they help to affect cellulose degradation. Okay, so with that, I hope I've convinced you that the Shipworm system is a system that really warrants further investigation. Just to summarize, it's different phylogenetically, functionally, and anatomically from anything else that degrades lignocellulose. It's simple. In the Shipworm symbiosis, we have 10 bacterial types doing the job that it takes 1,000 bacterial types to do in termites and ruminants. It's very rich in lignocellulose degrading enzymes, more than 1% of a protein-coding gene, so it's pretty easy to fish them out. It's highly specialized for cellulose degradation, so there's not a lot of other things going on there. It's unique in that it depends entirely on secreted enzymes. There's no contact between the bacteria and the substrate that's being broken down. And in that respect, it's more like some of the industrial bioconversion processes that are popular today. And finally, we think it's going to be really informative, because the shipworm selectively transports this complex cocktail of lignocellulose active enzymes from the gill to the gut. So essentially, it's telling us which proteins are important, right? It's the proteins that it sends to the digestive system. So now, instead of having to sort through millions of proteins present in a cecum or in a compost pile and try to figure out which ones are effective at working together, here we have the whole cocktail. And so this seems to be a really good place to look for mixtures of enzymes that could be useful for industrial processes. Okay, so with that, I just want to wrap up and give my thanks to all the people that I conned into working on this project, most without really adequate compensation. First, the folks here at Joint Genome Institute, Tanya Vojka and others who have just been fantastic help in this project. Our colleagues in the PMS ICBG program in the Philippines, our friends at New England Biolabs that helped us with the proteomics, our intrepid shipworm hunters, our colleagues at Oregon Health Sciences University, our friends at the KZ database who helped with the annotation, folks at the National Renewable Energy Laboratory who helped us with the carbohydrate assays, and finally my terrific crew back at Ocean Genome Legacy, postdoc Jennifer Fung and Cody Sharp. Jennifer's the lead postdoc in this project, and our technician Christine Reif. Okay, so with that, I'll be glad to answer any questions if you have them.
Yeah. You showed us very nicely that there are no bacteria in the cecum. What about small eukaryotes? Did you look for those?
I'm sorry, could you say that again?
Did you look for small eukaryotes in the gut in the cecum?
Small eukaryotes?
Yes, like fungi,
No.
like protists.
No, we don't see any eukaryotes.
Okay.
Yeah.
What do you think is happening to the other parts of the lignocellulosic material than the cellulose and hemicellulose?
The other parts, well, lignin we don't really know. That's sort of the most difficult area to study because the chemistry is not very well understood. And we have some sort of ambiguous data that suggests lignocellulose might be being modified, but I'm not happy enough with it to really say that anybody's really using it.
You said the shipworms can live on different kinds of diet. Do you see any difference in metagenomics or proteomics with different diet?
Well, that's something we hope to find out with our continuing work with the community sequencing program. So we're going to look at shipworms from a variety of locations around the world to see if we see those kinds of differences. Yeah.
A quick question, did you have to do anything special to get these symbionts to grow? Or was standard media, did you sort of get lucky? How did that work?
It's a story we don't fully understand yet, but it turns out that Bankia setacea is different from most other shipworms that we've examined in that it lacks pteridinobacter. It's the only one we've been able to cultivate these other strains from. So our current working hypothesis is that pteridinobacter is a bully, that it poisons or outcompetes the other bacteria. And in fact, my colleague Margot Haygood at Oregon Health Sciences University has been studying the secondary metabolites, the antimicrobial compounds that pteridinobacter produces. There's kind of a serendipity that that she started working on these, and it turns out that pteridinobacter is a really good producer of secondary metabolites. It has about 7% of its genome directed towards secondary metabolism, which is similar to some of the really important secondary metabolite producers like the actinomycetes. So that's our fantasy at the moment, and we think that that might be a really interesting story.
So you just use standard medium?
We just used the standard medium and had a lot of patience because these things grow very poorly.
Can you speculate as to why the gill is where you find all the bacteria and not the cecum? I mean, what's the evolutionary relationship?
I think it's an artifact. Well, let's say it's a historical accident. If you look at all bivalves that have symbionts, intracellular symbionts, they're always in the gill and they're always in the same cells. They're sort of homologous cell type. So most of the mollusk symbionts either oxidize hydrogen sulfide or they oxidize methane. I think that this symbiosis evolved by displacement of a sulfur oxidizing symbiosis. Because it makes a lot of sense to have sulfur oxidizers or methane oxidizers in your gill, because it's a place for gas exchange. But it makes no sense at all to have the cellulose degraders in the gill.
How are they passed to offspring?
We don't exactly know. There has been some data to suggest that there is vertical transfer for some symbionts, but I don't find the data very compelling. So we don't know.
Dan, is there a possibility that any of those bacterial-like proteins in the cecum are made by the host?
Yes, there is. We just finished the first draft of the Bankia setacea nuclear genome, and I haven't seen the data yet, so we're going to start looking to see if we find them there. But yes, certainly, eukaryotes can produce a small number of cellulases. Did I lose that? Oh, yeah, I'll just speak louder. The eukaryotes produce a small number of eukaryotes produce a small number of cellulases. So it is possible, and probably even likely, that the I should start dancing or something. Yeah, so it is possible that the host is producing some. Yeah.
So can you find the enzymes in the
No, we haven't found them there. We haven't looked, but a previous one of our collaborators has looked previously. Yeah.
And where inside of the gill cells are they? Are they in a compartment or a vesicle or?
Yeah, it's pretty interesting. They're in vesicles associated with the endoplasmic reticulum.
Okay.
So they're kind of like Legionella or Brucella, in that they're probably taken up by ER-mediated endocytosis. Yeah.
Okay.
You probably have some ideas for the transport of these enzymes into the cecum.
Yeah,
Did you look for motifs that are conserved across this small set of enzymes that you found?
We haven't found any of those yet, but there are some possible clues from the other proteins, the non-enzymes, that we see in the cecum fraction. So the only other types of proteins that we see are outer membrane proteins and some chaperonins. So I think it probably has something to do with maybe vesicle formation from the outer membranes. But beyond that, it's anybody's guess.
Thank you very much. I think Mario